Okay, so we left off last time finishing the action potential graph for a skeletal muscle. And the action potential graph that we just did actually applies to skeletal muscles and neurons. So we won't have to relearn it when we get to neurophysiology, but we will review it at that point. Okay, so we need that knowledge of how a muscle gets excited, in other words, fire and action potential, in order to understand the physiology we're going to talk about in this lecture. So we're going to start with a brief overview of some of the anatomy of a muscle, and then we'll go into it in a little bit more depth later on in this lecture. So a few terms I wanted to point out is that a single muscle cell is referred to as a muscle fiber. So if you hear us talking about muscle fibers, that's the same thing as just saying a cell. Some of the other anatomy I want to point out is the functional unit of a muscle is called a sarcomere. So what you're looking at down here is a single sarcomere. These are the contractile units of skeletal muscle, and they house the proteins actin and myosin. And they're arranged in a very specific way that gives us the structure of a sarcomere. And then the sarcomere itself, when they're all next to each other, creates these striations that you might have seen in skeletal muscle histology at the beginning of the quarter. So we'll be talking a ton about actin and myosin filaments today. Actin and myosin filaments arranged in sarcomeres are stacked next to each other or like arranged in a line in something called a myofibril. And these myofibrils, you can see here that there are many myofibrils per muscle cell. So we see that this whole thing is one muscle cell, and within that muscle cell, there's lots of myofibrils that contain these contractile proteins of actin and myosin. And then the muscle as a whole over here contains lots of muscle cells, which inside of them contain lots of myofibrils. So anyways, all this to say there are a lot of contractile proteins in skeletal muscle that allow our muscles to produce force. Something else I want to point out is the cell membrane on skeletal muscles is called the sarcolemma. The prefix sarco here means muscle. So you'll hear us, or you'll hear me talk about the sarcolemma. A lot of the organelles and anatomy of a muscle cell takes on the prefix sarco, and this applies to the plasma membrane, right, which we call the sarcolemma. The cytoplasm is called the sarcoplasm, and the endoplasmic reticulum is called the sarcoplasmic reticulum. So if you ever hear that word sarco, it means skeletal muscle. Okay, so let's zoom in on a sarcomere a little bit more. So this is one sarcomere. One sarcomere. And again, this is the structure that has myosin and actin arranged in a certain way that will allow us to produce force. So then here we can zoom in on the portions of myosin and on the portions of actin. So for myosin here, we have the myosin heads, and they're attached to like a long tail. And they're arranged in these like really thick bundles of protein. But it's particularly the heads of myosin that we are concerned with here that we'll be investigating today. Actin, on the other hand, is here in blue. You see it's this chain of proteins, and actin has these myosin binding sites, right? It's like these little donut-shaped holes here. So these are myosin binding sites. But what you might notice here is that on top of actin right now, there are two regulatory proteins that are blocking those binding sites. One is called troponin, one is called tropomyosin. And these need to be removed in order for myosin and actin to come together. So let's just kind of write this down to make sure we're keeping track of it. So contraction occurs when myosin binds and pulls actin. Troponin and tropomyosin, their job here is to block myosin. So you can imagine if we need to contract, if we need to do this up here, we need to get rid of troponin and tropomyosin. Now we may not need to fully get rid of these proteins, but we have to at least move them out of the way, right? Because they are blocking those binding sites on the actin. Okay, so now we can start to get into a little bit more of the anatomy, and what we're going to do over the next few slides is look at how a muscle cell contracts when it's stimulated by a neuron. So we have two pictures here. We have one really zoomed in, and here is our motor neuron. Right, like this whole thing is our motor neuron. The end of a neuron is called the axon terminal. So this portion right here is called the axon terminal. And then everything here is a muscle cell. This is a muscle cell. And then here, of course, are those myofibrils that have the sarcomeres in them. Right, so here it'd be like a sarcomere. Sarcomere. Let me change to black to show it better. This is a sarcomere. Okay, so just kind of arranging ourselves to the anatomy of a muscle here. There's a few other key structures that I'll talk about in just a second. Over here on the right, we see basically the same thing, but we have the motor neuron synapsing on the muscle cell, but the motor neuron is shown kind of as a whole. So the picture on the left is a really zoomed in portion to like just this bottom part here, right, where the axon terminal connects to the muscle cell. When we have this connection of like neuron to muscle, this connection is called a neuromuscular junction. This is called a neuromuscular junction, where a neuron and a muscle cell come together and they will communicate with each other. Okay, so what we're going to investigate is this neuron is going to fire an action potential. So there's going to be an action potential that comes down the length of this cell and it reaches the axon terminal. When that happens, the neuron is going to communicate this excitatory signal to the skeletal muscle. And then the skeletal muscle will then fire an action potential, which will travel all across the cell membrane. There's a few other structures you see on here that we haven't labeled yet. In green here, this is called the T-tubule. And then in yellow here, this is the sarcoplasmic reticulum. These are all very key structures. So that's the sarcoplasmic reticulum. All right, and then we're going to investigate like how these kind of structures are all playing together in the coming slides. So here we have a very similar picture. Again, on the left, we have the picture we were actually just looking at. And then over here on the right, we have the same picture, but actually slightly more detailed. So what I want you to do is maybe pause the video and see if you could label these structures on both of these pictures. Oh, I forgot to underline sarcomere there. So see if you could pause and label both of these on both of these pictures, label all of these structures. Okay, so on the left here, we have the motor neuron, which is over here on the right as well. Okay, so we got motor neuron. Let's do T-tubule. T-tubule is this part here. Right, the T-tubule is like this dip in the cell membrane, and the cell membrane like extends down into the cell to where it brings the cell membrane close to the sarcoplasmic reticulum. So this is the sarcoplasmic reticulum, also known as the SR. SR. So over here in yellow we have the SR and this webbing, and then in green we have the T-tubule. Okay, T-tubule, SR. Let's do sarcomere next. So sarcomere is going to be right here. And then similarly here, this is one sarcomere. I think, oh, mitochondria, last one. So mitochondria, they're just drawn here, mitochondria. And then over here, these are the mitochondria. So MT is the abbreviation for mitochondria. Okay, so what we're going to investigate is, again, how the motor neuron is actually communicating a signal to the muscle cell that causes the sarcomere to eventually contract. And you see that there's a lot of steps in between to get to the sarcomere contraction. So what we're about to do is the process of excitation-contraction coupling, and we have to start with the motor neuron. So whenever a muscle contracts, it always begins with stimulation from the motor neuron. So let's zoom in on the motor neuron and see what's happening there. So we have this diagram here, which has a motor neuron. And then we have a postsynaptic cell. This would be like the muscle cell. Muscle cell. And the little gap in between the two cells is called the synaptic cleft. And then there's a lot of channels that we need to talk about on this slide, and we'll bring them up as we get to them. So again, the first step of causing a muscle contraction is actually firing an action potential on the motor neuron. So this action potential will travel down the motor neuron and eventually reach the axon terminal, right? All of this is the axon terminal. Axon terminal. Okay, so within the axon terminal, we see that there are vesicles, and these vesicles contain neurotransmitter. The neurotransmitter that we're concerned with today is called acetylcholine, abbreviated ACH. That is acetylcholine, abbreviated ACH. So these vesicles are packaged with acetylcholine. All right, so what happens here is the action potential reaches the axon terminal, and this depolarization here, this positive charge that comes from the action potential, makes this region of the cell more positive. When that happens, it opens voltage-gated calcium channels over here, voltage-gated calcium channels. Once the voltage-gated calcium channels open, calcium starts to rush into these cells. So calcium rushes into these cells. And once calcium enters, it triggers exocytosis of vesicles. Of neurotransmitter here. So calcium comes in, exocytosis of neurotransmitter. This mechanism so far is actually extremely similar to the mechanism we looked at back in the integumentary system when we were looking at how melanocytes release the melanosomes, right? It was all coming from an initial depolarization, and now you kind of know more what that term means. The opening of voltage-gated calcium channels, calcium comes into the cells, and then it triggers exocytosis. So whenever calcium enters the cell, typically the next thing to happen is exocytosis of a vesicle. So we will exit, right? These vesicles will exit the cell. Maybe I should switch to blue here to keep it consistent. And now we have lots of acetylcholine present in the synaptic cleft. So this is all ACH in the synaptic cleft, that little space, fluid-filled space in between the two cells. Acetylcholine is going to diffuse across this space and eventually reach the muscle cell on the other side. And on the muscle cell side of this is where we have our ligand-gated ion channels. These are the receptors for neurotransmitter. And the specific neurotransmitter we're looking at are ACH receptors. So these are all ligand-gated ion channels for ACH. Which means they have these little binding domains, right, for ACH. So let's say ACH binds to the ligand-gated ion channels and it opens it. The ion that's going to come in is then sodium. So sodium will start to enter the muscle cell here once acetylcholine binds to these receptors and opens them. And now what we've done is we've increased the amount of sodium present here, and we've increased the charge on the cell membrane. This is going to eventually lead us to action potential across the rest of the muscle cell. Action potential across the rest of the muscle cell. Okay, so one more thing to point out on this slide, it's actually not drawn in on here, But if we have acetylcholine present in the synaptic cleft here, it is just going to keep stimulating the skeletal muscle until the acetylcholine is gone. And if we don't have any way to get rid of acetylcholine, muscles will just stay contracted. So we need a way to get rid of acetylcholine. And we do. On the side of the muscle cell, we have these proteins, these enzymes, that actually break up acetylcholine. So acetylcholine could bind to them. And these enzymes here in purple, so let me just make like a little legend over here. So these purple dots are an enzyme called acetylcholine choline esterase. Esterase. The abbreviation is ACHE, acetylcholine esterase. Its job is to break down acetylcholine. So remove acetylcholine from the synaptic cleft. If acetylcholine is removed from the synaptic cleft and the motor neuron stops firing action potentials, then we will stop having muscle contraction because there's no more acetylcholine to stimulate the muscle cell. Okay, so that's the events. Everything we just did, these are the events at the neuromuscular junction. Okay, so we have the events of the neuromuscular junction, and then now we have to introduce the rest of the muscle cell and then what's happening in the sarcomere. So I have this drawing for you that I made myself of the neuromuscular junction and then the rest of the process. So what we're going to do on this slide, again, is I mentioned it slightly earlier, but we're going to go through what's called excitation, contraction, coupling. So this is the process, also called EC coupling in skeletal muscles. So this is the process of the skeletal muscle being excited and then resulting in a muscle contraction. So why don't we actually just review what we did on the last slide, right? We have the neuromuscular junction, so let's just draw that back in. So that action potential reaches the axon terminal. Then we get this exocytosis, right? Exocytosis of ACH. And over here, we have these ACH receptors. Once ACH binds to these receptors, sodium can then enter. Sodium enters. And the cell membrane starts to depolarize. And that depolarization is going to travel like a wave down the cell membrane of skeletal muscle. And over here, as this action potential is traveling as a wave down the cell membrane, this is where we have these cells having an action potential. So you know the graph that we drew last time, right? That looked like this. It was at rest, then it had this action potential, and then it came back to rest, right? This is our action potential graph. This graph is happening like at every single point along the skeletal muscle membrane, all the way down until we get into the T-tubule. So every single portion of the cell membrane needs to have this action potential in order to move this wave of ions down the cell membrane. So what's actually on the cell membrane here? Well, it's the same channels that were found in our action potential graph. So it's actually a good review of our action potential graph. So let's see, maybe we can use two colors here. So in green, I'll put them kind of spaced out. In green here, we have voltage-gated sodium channels, right, responsible for that depolarization phase of our action potential. So when they open, sodium would come in to these cells. And then we have to repolarize. So maybe in red here, in red, we have voltage-gated potassium channels. And when these open, potassium would exit the cell. And that the potassium part would be down here. Okay, so we have action potentials traveling down the length of the cell membrane, and it's eventually going to dip into the T-tubule. The T-tubule has one really important protein on it. This protein is called the DHP receptor. This is a voltage-gated channel, So it opens in response to this wave of depolarization, right? This action potential traveling down the T-tubule. And of course, like I just drew the action potential in the middle of the T-tubule, but the action potential is a membrane event. So it'd be happening on the actual membrane. But just for simplicity, I drew it kind of like inside the T-tubule. But when the action potential reaches the T-tubule, it will open the DHP receptor, right? Because it's just voltage gated, so it responds to a change in voltage. And then the DHP receptor, this part is really interesting. The DHP receptor is like physically tethered, physically connected to a channel on the SR, right? So this is our sarcoplasmic reticulum. So it's physically connected to a channel on the SR. This channel on the SR is called the ryanodine receptor or the RYR receptor. So we'll just refer to it as the RYR receptor. But the full name of it is ryanodine receptor. The ryanodine receptor, RYR, is a mechano-gated calcium channel. So when the DHP receptor opens, it is physically connected to the RYR, and it kind of, if you will, pulls on the door of the RYR. And this causes a mechanical deformation of the cell membrane, of the sarcoplasmic reticulum membrane, which opens this mechanogated calcium channel. Once calcium channel is open here, the calcium inside of the SR can now flood out. So from this slide, we're actually seeing the SR for the first time in a little bit more detail as well. And we see that what is happening in the SR is it's really just a storage unit for calcium, right? So the SR is just a storage unit for calcium. So if we open the door on the storage unit, the contents can just spill out, right? They can start to exit. So we start to get lots of calcium present in the cytoplasm because it exited the SR into the cytoplasm. Now that we have calcium out, calcium will make its way to the sarcomere. So we have zoomed in on a sarcomere here. This is a zoomed in sarcomere. And you might remember from a previous slide that the key proteins we have in a sarcomere is the actin, myosin, and then on actin we have those two regulatory proteins, troponin and tropomyosin. And earlier I told you that they are blocking myosin from binding to actin. So if we want myosin and actin to come together, we have to remove troponin and tropomyosin. And that's exactly what calcium does. The role of calcium is to remove troponin plus tropomyosin off of actin. It doesn't completely remove it, it just shifts it slightly so that it's no longer blocking those myosin binding sites on actin. So calcium will come in, bind to troponin. This causes the troponin-tropomyosin complex to move off of actin. And now that troponin and tropomyosin have slightly moved away, myosin is now free. Myosin can bind, can bind actin. Okay, that slide was a lot, right? And over the past couple slides, we have introduced just a ton of new content, starting all the way at the motor neuron up here. So this is the entire process called excitation-contraction coupling. And here is the really nice textbook drawing. If you would like to look at it, it has all of the same events that we just talked about, and it has the same receptors, right? Some things to point out. We have acetylcholine, we have the ryanodine receptor, the DHP receptor, and you see that like physical tether between them. So that when the DHP opens, it like physically opens up the RYR. And then calcium is released, right? Calcium is released, which can then bind to, right over here, calcium binds to troponin, allowing actin and myosin to bind. This is what we're trying to get to, actin and myosin to bind. Okay, now that actin and myosin are free to bind with each other, this is where we finally get the force production step. So what we're going to do on this slide is something called the crossbridge cycle. Crossbridge cycle. Now, to understand the crossbridge cycle, a few things. One, it is a cycle, so there really isn't a single starting point. Like, myosin can start at any one of these points. If you look at different pictures in different textbooks or on the internet, you're going to see a slightly different starting point every time. Because it's a cycle, we can start wherever. The one thing we do have to do, though, is have that calcium signal come in to remove troponin and trypomyosin. So again, like no matter what picture you're looking at, in order for muscle contraction to happen, we have to first have calcium present to remove troponin and trypomyosin. Okay, but let's just focus on actin and myosin for this slide. And for myosin, myosin actually has, this is myosin has like an affinity list. So you might remember the concept of molecular affinity from previous bio classes, right? It's how likely two things are to interact with each other. So if two proteins have a high affinity for each other, they will interact with each other. And it's not just proteins, right? It could be anything. It could be sodium moving through a sodium channel. It could be ATP binding to an enzyme. So myosin has a preference for what it's binding. And the first on its list is ATP, and the second on its list is actin. And it can't bind to both actin and ATP at the same time. So if ATP is present, myosin is going to bind ATP and not bind actin. But if ATP is not present, myosin is going to bind actin and not ATP. So it can only bind one at a time, and it prefers ATP. So let's start up here. We'll just say this is like step one. We have myosin currently just bound to actin. So like let's say it just finished one cycle. It just finished a muscle contraction. What happens next? ATP comes in and binds to myosin, right? So ATP binds to myosin, and then myosin releases actin. So myosin releases actin. Remember, it prefers ATP. So if ATP is present, it's going to bind that and release actin, which is exactly step one here, right? Binds ATP, releases actin. The next step is myosin is actually an enzyme. It can go through ATP hydrolysis. So it can actually hydrolyze ATP into ADP plus inorganic phosphate. And when it does that, it kind of cocks forward or moves forward into this like ready position here. So that's kind of what you're seeing here, right? This is like the future of where myosin is going. It hydrolyzes ATP into ADP and is now kind of moved forward. But here's the thing, ADP is no longer ATP, right? Those are two different molecules. So myosin goes down its affinity list and says, oh, well, actin is here, so I'm going to bind that. Myosin then binds to actin. It has ADP still bound. Don't worry, it's about to let go of it. It has ADP still bound, but it is now attracted to actin because it has a high affinity for actin compared to ADP. Okay, now it is going to release ADP. So it releases ADP plus inorganic phosphate, and this pulls actin, or it pulls myosin back to its original position, but because myosin is holding on to actin, it also pulls actin. And this is called the power stroke. So this is where myosin is pulling on actin. This is force production. Force production. Okay, and myosin is now bound to actin, but it's let go of ADP and inorganic phosphate, and it has just now finished a cycle. So it just finished contraction. What happens next? A new molecule of ATP comes in, binds to myosin. Myosin hydrolyzes it, goes into this high energy position, this cocked position. Once it hydrolyzes ATP, it rebinds to actin, right? Rebinds actin, and then it pulls. Let's go of ADP and PI, bound to actin, just finished muscle contraction, new molecule of ATP, hydrolyzes, rebinds to actin, pulls. And it just keeps repeating this process, right? So this is a cycle. One note here, right? It has like ADP and PI technically releasing at different times. Some textbooks will show it slightly differently. I don't really care if you know when ADP and PI releases because it's not like that important to me for the crossbridge cycle. I would rather you know the major steps of this, which is uh, ATP binding to myosin causes it to release actin, and then it hydrolyzes it forward, binds actin, and pulls. And then it just repeats that process. And I actually have a picture from your textbook that I think is a really helpful like analogy and visual of what myosin is doing here. Right, so myosin first grabs the rope here, crossbridge formation, and then it pulls. This is force production, but then it has to let go, right? Myosin has to let go, move forward, and then pull again, like grab and pull again. So I have a little brain exercise for you, right? Thought exercise for you on this. Where on this diagram would ATP come in? So when is ATP binding to myosin and when is it being hydrolyzed? So let me just write that down. When on this diagram is ATP binding to myosin and when is hydrolysis occurring? Right, so like the grab rope, the pull, the let go, the reach forward, which two of these refers to binding and then hydrolysis? Okay, we just covered a ton of material, so why don't we do the flow diagram for excitation-contraction coupling just to make sure we're all on the same page. And this will be one nice review slide for us to easily be able to recall this content. Well, it may not be easy at first, but this will be the tool that you're going to use to then make it easy to recall. So again, we haven't done a flow diagram in a little while as we were in bones and joints. So the last time we looked at a flow diagram was kind of in our first bone lecture, right? When we looked at like bone remodeling. And then I also introduced flow diagrams in our melanocyte lecture, right? In the integumentary system. So again, just a reminder here that a flow diagram is a cause and effect relationship that shows a biological process. So we're going to do that in excitation-contraction coupling. And this is a beast of a flow diagram. Let's do it together. Okay, so we're going to start with action potential reaches axon terminal. Actually, if you want to try to pause and try to do it yourself, you can. I'll give you the starting and stopping steps. Can you go from action potential reaches axon terminal, the cause and effect, all the way down to muscle contraction? Now, a few things here. Somewhere down here, you will talk about the crossbridge cycle. And again, the crossbridge cycle is a cycle, so you can kind of start it wherever you want. It's likely that when I start to talk about the crossbridge cycle, it's going to look slightly different from when you start to talk about the crossbridge cycle. So just depending on where you start, maybe you just talk through like one crossbridge cycle, right? Like one full cycle of crossbridge formation. So if you would like to pause and test yourself here, reviewing the previous slides, you can. Otherwise, let's do it together. Okay, so action potential reaches axon terminal. This opens, right, causes the opening of voltage-gated calcium channels. Voltage-gated calcium channels, right, opening in response to the depolarization. If voltage-gated calcium channels open, this causes calcium to enter. Calcium enters. Axon terminal. Okay, calcium's role in the axon terminal is to trigger exocytosis. Exocytosis of, just a reminder that our neurotransmitter here is called acetylcholine. So it triggers exocytosis of acetylcholine. Acetylcholine then binds to ACH receptor on skeletal muscle. So ACH binds to receptor on skeletal muscle, and this opens ligand-gated ion channels. So opens ligand-gated sodium channels. So if sodium channels are open, then sodium can enter. Now we're on the muscle cell, by the way. So sodium enters muscle cell. Okay, this sodium entering the muscle cell causes a depolarization across the sarcolemma. Remember, sarcolemma is just the cell membrane of skeletal muscle. That depolarization will eventually enter depolarization. So depolarization enters T-tubule, or travels right across the T-tubule membrane, and this opens the DHP receptor. The DHP receptor is physically tethered to the RYR, so when the DHP receptor opens, this causes the opening of the mechano-gated RYR. I'm just going to put here on SR, just to help us remember where the RYR was situated. If the SR is now open, then calcium exits SR to cytoplasm. Now that calcium is in the cytoplasm, calcium binds troponin and this removes the troponin-tropomyosin complex. I'm going to abbreviate troponin and tropomyosin here. So TN is troponin, TM is tropomyosin. So it removes the troponin-tropomyosin complex. Oof, this is a lot, right? Removes troponin-tropomyosin complex. Now we can finally do crossbridge cycle. I might move this slightly up here just to give myself more room. So removes the troponin-tropomyosin complex. Now we have the crossbridge cycle. So then myosin. Why don't we do one more step here before we get into the crossbridge cycle? We'll say myosin binding sites revealed on actin. Okay, now myosin can bind to actin. So myosin binds actin, which causes it to pull actin. And this is the power stroke. Power stroke. After it pulls on actin, it needs to bind ATP. Right, new molecule of ATP comes in, binds to ATP, which causes the release of myosin from actin. Myosin hydrolyzes ATP. Myosin rebinds actin. Right, so myosin binds actin. Then we get to muscle contraction. Right, so muscle contraction here is just kind of like this just keeps happening. Keeps happening. Okay, so there is the flow diagram for excitation-contraction coupling. This is one of the longest flow diagrams in physiology, but I hope this helped because this material is so dense. So again, I'm trying to equip you with a tool that you can use to study a complicated process in a more structured way. Okay, maybe this can be the last slide because this has already been a very dense lecture. We've just talked a ton about relaxation at the molecular level. Why don't we now, sorry, we just talked about contraction at the molecular level. Why don't we now talk about what happens to relax? Don't worry, it's a lot faster than contraction. So in order to relax the muscle at the cellular molecular level, a few things have to happen. The main thing is that the motor neuron needs to stop firing. So motor neuron stops firing. Action potentials. Stops firing action potentials. Once it stops firing, the acetylcholine will no longer be released by the motor neuron. It will eventually get broken down by acetylcholine esterase. So you remove this excitation portion of the process. The DHP and RYR are now closed, but calcium is still present in the cytoplasm. So if calcium is still present, it can technically still bind to troponin and tropomyosin. And calcium is binding and unbinding all as random events, right? Everything in biology is happening via random events, and things are coming together if they have high molecular affinity. We have another protein on the surface of the SR that I haven't told you about yet. It is called the calcium ATP ACE, or the calcium pump. So the calcium ATP ACE will move calcium from the cytoplasm back into the SR. So this is the protein that creates and maintains the calcium gradient, which is always high in the SR, low in the cytoplasm. So once the calcium has been removed from the cytoplasm back into the SR, the motor neuron is no longer firing, there's no more acetylcholine, all the channels close, troponin and tropomyosin can rebind, right, recovers actin, and myosin can no longer bind to actin. And that's relaxation. Okay, good job with this difficult lecture.